The genetics of deafness is very complicated. There's right now over 150 different genes that, when mutated, can lead to hearing loss.
Dr. Lawrence Lustig is chair of Otolaryngology, head and neck surgery at New-York-Presbyterian and Columbia. He has spent decades working to advance therapies to treat genetic hearing loss. Historically, this type of hearing loss could only be treated with cochlear implant surgery. But Dr. Lustig was determined to find a better way. He thought that gene therapy, delivering the gene that is missing in people with genetic hearing loss directly to these patients, could be the answer.
In my initial kind of thought process, I was thinking, I can take a kid and not have to put an electronic device in their skull for the rest of their life.
In pursuit of that goal, Dr. Lustig's lab at New-York-Presbyterian and Columbia was the first to show that gene therapies could effectively reverse genetic hearing loss in mouse models using a glutamate gene called vGlut3.
Glutamate is the major neurotransmitter of one of the hair cells of the cochlea, the inner hair cell. It's how the hair cell talks to the auditory nerve. I think the vGlut3 study that we did on the mice, that's really the first time people saw that you can take a mouse model and totally restore the hearing.
After this breakthrough, he and his team faced a new challenge. Would this same mechanism work to restore natural hearing in humans with genetic hearing loss?
There was a gut feeling in everybody who was involved in this project that it was going to work. It worked in the animals. It's got to work in people. But at the end of the day, you do not know until it goes into somebody and you see what the results are.
After years of research and clinical trials, in April 2026, the FDA approved the first ever gene therapy for genetic deafness based on the work pushed forward by Dr. Lustig and his team.
When we started seeing the hearing results get better and better and better, it was jaw-dropping. None of us can believe how well these kids were hearing. To me, this has been the most gratifying stretch of my entire career, watching this whole thing unfold.
I'm Erin Welsh, and this is Advances in Care. Today, I'll speak with Dr. Lawrence Lustig about his groundbreaking gene therapy research to restore natural hearing in patients with genetic hearing loss. Dr. Lustig, thank you so much for joining me today.
It's wonderful to be here. Thanks for having me, Erin.
So to kind of set the stage here, historically, it seems that hearing loss has been difficult to address at the genetic level. What are some of the reasons for that? And when did you realize that gene therapy could be a potential solution?
Once your hair cells die, they don't regrow. So there's been a lot of efforts around the world to try to get hair cells to regrow. I think the vGlut3 study that we did on the mice, that's really the first time I think people saw that you can take a mouse model and totally restore the hearing. The problem was vGlut3 is not a common cause of deafness. And that's why we started going for more common causes of genetic deafness, where we thought there really was good gene therapy applicability.
So I understand that one of the first genes you looked to target was otoferlin, which is a more common cause of genetic deafness. Tell me about why this seemed like the right gene to look at in terms of keeping your eye on the goal of eventually trialing this therapy in humans.
So the vGlut3, it's a synaptic protein. It involves the neurovesicular release of the neurotransmitter onto the neuron that goes to the brain. And there's something about that specific kind of mutation that does not lead initially to hair cell death or any other cellular death. So we thought synaptic proteins are going to be the way to go here, because a lot of the other mouse models we looked at, there was a lot of cellular damage. So we said, okay, otoferlin, it's a known cause of deafness. It creates what we call an auditory neuropathy because the specific mutation on the otoferlin gene basically causes it to make a protein that doesn't work, right? It's a null mutation, meaning there's no function of the gene. The otoferlin gene allows the synapse to release the neurotransmitter onto the afferent neuron, which goes off to the brain. Without otoferlin, glutamate cannot be released, and it's like flipping off the light switch. And all you need is otoferlin back in the mix, and the synapse works normally. This was a target because it was a synaptic protein, because there's good cellular preservation. We did get a hold of the animals and studied them, and everything looked good. And we said, this is going to be perfect. The problem that we had was that otopharylin's a rather large gene. It's about 6.5 kilobases, whereas the viruses that we typically like to use for gene therapy for the ear, adeno-associated viruses, they have a maximum packaging capacity of about 4.5 kilobases.
Okay, so you had a problem of size on your hands. How did you address that challenge?
We had tried a number of things. We tried chopping the gene up into little pieces. We tried making these mini otopharylins, and we didn't get any hearing recovery. There was a bust. And then, early on, I had read about this new approach for larger genes called a dual vector approach. And one of the papers I had read, they had taken this large gene called myosin 7A, which is involved in muscle function, and they cut it in half, and they engineered sticky portions on each half. So when you took and loaded each of those halves into a separate virus, and when you co-inject those viruses into a cell, the sticky portions find themselves, right? And then they cut themselves out. And now you're left with the full-length messenger RNA, which makes the full-length protein. It's brilliant. I loved it.
That's brilliant. So it's like we're taking a book, we're splitting it into part 1 and part 2, we're delivering them separately, and then you put them back together with Velcro, essentially.
Yeah, but then the Velcro gets cut out itself.
And so then there's no trace. So there's no trace. Here's the full delivery of this
exactly.
Gene.
So it turns out one of the authors on that paper was the guy that was making all of our viruses for all of our other gene therapy studies. He made us the vectors, and then we tried those vectors, and it worked. And the mice heard almost completely normally again. And the big difference between this one, so when we did the VGLU3s, if we delivered it about 12 days after birth, which is still considered like being in the uterus, right? We got really good hearing results. But if we did it at 21 days after birth, we didn't get any hearing results at all. So there's something that happened with the maturing ear that it didn't seem to work very well. But with the otopharylin delivered later when the mice were older, they heard even better. Because 30 days after birth, now you're talking about infancy within the first 6 months of life. So now you've got a real potential candidate that you can move into humans.
That must have been so exciting. And so tell me more about how the biological mechanism behind the split vector approach worked in practice. Like, why were these results so promising?
Well, I think you've got this construct that's cranking out your natural protein. It's getting to the right location. It's doing what we want it to do. We don't have worries about what's called insertional immunogenesis, meaning the gene getting into another gene and causing a problem. This is a virus that doesn't cause human disease. It seems to be extremely well tolerated. We really saw no gene therapy related side effects. Another really great thing about this therapy is, all genes have a region of DNA called a promoter, which tells the gene when to turn on and when to turn off. And we're using a hair cell specific promoter for the gene therapy. So even if the virus were to inject the DNA into a cell that doesn't make otopharylin, it wouldn't turn on. So there really doesn't seem to be any side effects that we've been able to detect.
So we kind of talked about targeting this specific gene, developing this gene therapy, and then how do we get from having this gene delivered by virus into animals to then envisioning this in humans? What were the steps required for that process?
Oh, that part's easy.
Oh, I'm sure.
So I think this. I will say, the fallacy is to think, oh, I've got this great idea. Let's just get into people, right? Then the climbing really starts, right? There are tons of preclinical studies that you have to do to make sure that it's safe. And you've got to define how you're going to do this very clearly to the FDA. Then you have to figure out, what's the concentration of the drug that you're going to use, right? How are we going to get this in the ear? We decided to go through the mastoid bone like we do for a cochlear implant. We got to figure out the dosage, the concentration, which age range are we going to do? So these were countless hours of meetings with groups of docs like myself from around the country who were figuring out exactly how we were going to run this clinical trial. We published on the VGLU3 knockout what caused the hearing loss, I think around 2008. We did our 2012, we actually published the data on the hearing restoration in that mouse. And it wasn't until 2018 that we published the data on the otopharylin knockout mouse. To me, that's a shockingly fast timeline to go from publication in a mouse model to getting into humans within about 5 or 6 years.
Yeah, it's almost unheard of.
Yeah.
It's amazing. Oh. So can you walk me through the steps that you took to administer the gene therapy during the clinical trial?
The approach we take is pretty much identical to how we do for a cochlear implant. We make a little incision behind the ear. We drill through the mastoid bone. That gets us into the middle ear. We make a very tiny opening through this little membrane called the round window membrane, put in a little catheter. We also make an accessory opening into the balance canal, because this allows us to actually flush the whole cochlear fluid and really make sure we get good delivery. And then we just flush this drug in very slowly through a micropump for about 15 minutes. We close everything up, and then they go home. But it's not like a cochlear implant where they go home and then 3 weeks later we turn on the implant and everybody's seen those videos. The kids light up. Well, this is not like that, right? It's like the ear slowly wakes up over the course of 6 to 12 weeks. We know from studying kind of auditory development in animals that, the hair cells are actually firing nonstop during development and patterning the auditory nerve. So that hasn't happened. So now all of a sudden this hair cell's it's firing this neurotransmitter, and the nerve, which has gone through all of development with hearing nothing, it starts waking up. Imagine like when you fall asleep on your arm and your arm's kind of dead, right? And you open it and you kind of shake your hand and all of a sudden you can feel again. Well, imagine that process happening over months, right? So the nerve has to wake up. Think about all the pathways from the inner ear all the way to the auditory cortex. Those all have to wake up. Those all have to start patterning again. And at one point, I remember the mom calling me and saying, "Oh my God, my kids are running out of their house screaming." Or someone turned their head to something that dropped on the floor. These unexpected things. And then to watch the hearing gradually get better and better and better.
I mean, that is so exciting that during the clinical trial, you saw results like these coming in, and it really showed what an impact this could have on people. And so can you tell me more about the role that New York-Presbyterian and Columbia played in developing, launching, and executing the clinical trial?
There's lots of regulatory activities that have to happen to get approval for a clinical trial. But having been involved in the entire planning phase, I think we were ahead of the game. We were one of the first sites that was online. Then it was just trying to find the kids, right? That's one of the great things about being in a large academic medical center like New York-Presbyterian. You've got an amazing patient population that draws patients from not only all over the country, but all over the world. We had been marketing and talking to everybody about this. And, if you have an otopharyngeal kid, please send them to us. And so we actually had 3 kids identified very early on. In fact, we did more kids at our center than any other center in the U.S. because I think we were so early on the ball on this and outreaching so extensively. I think that's what sort of helped get these 3 kids in here. And, the first family was an older sister and a younger brother. And the family was not interested in a cochlear implant. They didn't really feel that was their pathway. They were going to send these kids to deaf school. But then they heard about the gene therapy. I reached out to them when I heard about the mutation. I was hooked up through their otolaryngologist up in upstate New York. And then the family decided they want to do it.
How old were these 2 kids?
They were about 2 and 4 at the time of dosing.
And in general, what is the age ranges that you're targeting with this gene therapy?
My original thinking was that if you don't get to these kids by 6 or 8, you're not going to get any benefit whatsoever. But in fact, what we've seen now with some of the other studies, and even people dosed as late as 21, 31 years of age, still getting some auditory benefit. The window with which we can intervene is actually much wider than we thought. But I still think that the earlier you can intervene, the better. And this is data that we know from cochlear implants. So the earlier we can put in a cochlear implant in a deaf child, the better the results that they're going to do. I think it still is about early diagnosis and early intervention.
So a lot of these kids, though, might be too young to be able to communicate their feelings clearly, but the families witnessing this transformation, what is that like for them?
It's unbelievable. One of the moms of the more recent kid that we dosed, the other child at around 2 years of age too, showing him dancing and around the house. And, she sent me a video talking about, how he's going to hear his girlfriend say yes when he asked her to the prom. She's, it still brings a tear to my eye, Yeah, it's kind of incredible. You can get that with a cochlear implant, right? You can get hearing and language development, but it's not natural hearing.
Right.
It's not natural hearing, and you have to manage this device in your skull for the rest of your life, and to have like a single shot injection of a drug in the ear that we think will last a lifetime. I mean, that's kind of amazing.
I know that this is now an FDA-approved therapy. What are some of the results that you're seeing so far in terms of response rate?
Most people number one, if you respond within the first 6 weeks, your hearing is going to probably be some of the best. We're seeing hearing in the normal range all the way to the severe range. Every study has one or two candidates that hasn't responded at all, who've gone on to cochlear implants and have done as well as any cochlear implant child has done. So there doesn't seem to be a downside of doing the gene therapy in terms of being able to benefit from an alternative therapy if it doesn't work. Interestingly, if you look at how well one ear does in the bilateral kids, meaning both ears, the other ear tends to do the same, meaning there must be some basic molecular background, genetic background that sort of determines how well an ear is going to do. It doesn't seem to be technique related or anything like that in terms of the delivery. And I think the real fun is going to be when these kids are much older. And the really fascinating kids are going to be the one with an implant on one side and the gene therapy on the other. Then you're really going to be able to understand what's the difference between an implant and natural hearing in those kids.
Yes. Oh, that'll be really interesting to see.
Yeah.
What are you most excited to see this research accomplish in the next, say, decade or so?
This is an amazing success, and it's a proof of concept that we can cure genetic deafness. Now, the real question is can we apply this to other forms of genetic deafness, much more common forms, things like connexins, which can account up to a quarter or a third of cases of genetic deafness out there. Each of these other forms have challenges that we didn't have to overcome with otolaryngology, but there's a number of candidate genes that are on the chopping block right now. Connexin's one of them. For connexin, I know we're going to start seeing clinical trials within the next 1 to 2 years. But for these other ones, within the next 3 to 5 years, we're going to start seeing clinical trials to see who's a candidate, who's going to benefit. Can we, particularly for those forms of deafness that come in later in life, and we can identify them, can we intervene earlier and prevent the hearing loss that would otherwise occur and make them cochlear implant candidates? And I think that's another big area of promise. It just makes you hopeful that the next 30 years is going to be equally transformative.
I'm certainly hopeful after speaking with you. It is incredible to think about where this work might go. Dr. Lustig, this has been such a inspirational, thought-provoking conversation. I really, really enjoyed discussing your work with you. Thank you for joining me.
I've had a great time today, Erin. Thank you so much for having me on your show.
Thanks so much to Dr. Lawrence Lustig for speaking about his truly groundbreaking work using gene therapy to restore natural hearing in patients with genetic hearing loss. I'm Erin Welsh. Advances in Care is a production of New York-Presbyterian Hospital. As a reminder, the views shared on this podcast solely reflect the expertise and experience of our guests. To listen to more episodes of Advances in Care, be sure to follow and subscribe on Apple Podcasts, Spotify, or wherever you get your podcasts. And to learn more about the latest medical innovations from the pioneering physicians at New York-Presbyterian, go to nyp.org/advances.